Welcome. I am so glad we are sitting down to look at this topic together because it is truly one of the most fascinating mechanisms in all of biology. Today, we will explore exactly how our cells make split-second decisions to keep us alive using protein kinases and phosphatases. To start, imagine a massive factory filled with thousands of workers, which represent the proteins inside your cells. These workers are just standing around doing absolutely nothing until they are handed a very specific glowing ID badge, which represents a phosphate group. This simple, beautiful act of adding a tiny chemical tag is exactly how cellular machinery is turned on and instructed to perform its specific job. Without this system, the factory would be completely frozen. The workers have the potential to do the work, but they lack the authorization. Now that we have a picture in our heads, let us look at the formal scientific definition you will see in your textbooks. The manager who walks around handing out these badges to turn the workers on is called a protein kinase. In molecular endocrinology, a protein kinase is a specialized enzyme that catalyzes the transfer of a phosphate group from an ATP molecule to a target protein. This vital chemical process is known as phosphorylation, and it is the primary way cells transmit signals internally. Building on that definition, it is important to note that kinases are highly specific in their actions. They do not just throw phosphate groups anywhere on a target protein. Instead, they target very specific amino acids to ensure the correct structural change occurs. In most cases, phosphorylation specifically targets the amino acids serine, threonine, or tyrosine. By attaching the phosphate to these exact spots, the kinase ensures the protein receives the correct signal and performs the exact function required by the cell at that moment. While kinases turn things on, we also need a reliable way to turn them off. The strict security guard who walks around taking the badges away to turn the workers off is called a protein phosphatase. Conversely to kinases, a protein phosphatase is an enzyme that removes this exact same phosphate group from the target protein. It does this through a water-based reaction called hydrolysis, which we call dephosphorylation. This removal is just as important as the addition, ensuring signals do not last longer than necessary. Together, these two opposing enzymes act as the ultimate molecular switches within the endocrine system. When a phosphate group is added or removed, it physically alters the three-dimensional shape of the target protein. Because structure always determines function in biology, this is a profound event. When the protein changes shape, its function, its overall activity level, or even its physical location within the cell changes instantly. This allows the cell to react to external hormone signals in a fraction of a second. You might be wondering why we need such a complex system of adding and removing tags. Without kinases and phosphatases, the hormone signals in your body would either never start or they would never, ever stop. For example, when insulin binds to your cells, it activates specific kinases that tell the cell to rapidly absorb glucose. If the phosphatases did not eventually remove those phosphate groups, your cells would keep absorbing glucose until your blood sugar dropped to fatal levels. This delicate balance is so critical that when it breaks down, the consequences for the body are severe. In fact, hyperactive kinases can tell cells to divide uncontrollably, which is a primary hallmark of cancer. Because of this, many modern medical miracles, including highly advanced cancer drugs, are actually kinase inhibitors. These drugs are specifically designed to block those hyperactive kinases, stop the runaway signaling cascade, and restore order to the cell, proving just how vital this tug of war is to human health. To understand this tug of war mathematically, we use the steady-state phosphorylation formula. This equation shows us exactly how much protein is active at any given time. P star represents the concentration of the active phosphorylated protein. P total is the total amount of that protein available. The symbol Kk is the rate constant of the kinase, showing how aggressively it adds phosphates. The symbol Kp is the rate constant of the phosphatase, showing how fast it removes them. If the kinase rate increases due to a hormone, the active protein goes up, but if the phosphatase rate increases, it goes down. Let us see this formula in action by calculating the baseline state of a cell before any hormones arrive. We're given a total enzyme concentration of 100 nanomolar. The background kinase rate is 2 per second, while the phosphatase rate is much higher at 8 per second. We plug these values into our steady-state equation. The numerator becomes 200, representing the kinase drive, and the denominator becomes 10, representing the total system turnover. Dividing these gives us a final answer of 20 nanomolar. This means in the resting state, only 20% of the enzyme is active, preventing the cell from wasting energy. Now, suddenly, a hormone binds to the cell, activating a signaling cascade. This increases the kinase rate dramatically to 42 per second, while the phosphatase rate and total protein remain unchanged. We use the exact same formula, updating only the kinase rate. Our numerator is now 4,200, reflecting a massive push by the kinases. The denominator grows to 50. Dividing these gives us a new active concentration of 84 nanomolar. By simply increasing the kinase activity, the hormone has successfully flipped the cellular switch, activating 84% of the proteins to carry out the biological response. To visualize everything we have calculated, let us look at the phosphorylation cycle diagram. On the left, we have the inactive protein. Following the top arrow, you can see the protein kinase using ATP to attach a red phosphate group, converting it into the active protein on the right. Following the bottom arrow back, the protein phosphatase uses water to remove that phosphate, returning the protein to its inactive state. This continuous loop is the exact dynamic tug-of-war we just modeled mathematically. To wrap up our session, I want you to remember that protein kinases and phosphatases are the ultimate molecular switches of the endocrine system. Kinases turn signals on by attaching phosphate groups, while phosphatases turn them off by removing them. Understanding this delicate mathematical balance is the absolute key to understanding how hormones control everything from your metabolism to cell growth. You have done a great job mastering this continuous, dynamic tug-of-war.